The overall goal of this assay is to enumerate and characterize Simian immunodeficiency virus-specific CD8 positive T cell populations generated by vaccination or primary infection.
This method can help answer key questions in the T-cell immunology field, such as what is the frequency of antigen-specific CD8 T cells induced either by vaccination or infection. The main advantage of this technique is that it can quantify antigen-specific CD8 T cells in biological samples obtained directly ex vivo without the need for in vitro re-stimulation.
The implications of this technique extend toward the evaluation of antigen-specific CD8 T cell in the Rhesus macaque and for studying CD8 T cell responses in multiple disease settings. Generally, individuals new to this method will struggle because of the incomplete knowledge available for Rhesus macaque MHC genetics, and because the flow cytometric staining requires precise antibody application.
Begin by resuspending the Rhesus PBMC in R10 medium at a 1.6 times 10 to the 7th cells per milliliter concentration. Aliquot 50 microliters of cells to the appropriate number of flow cytometry tubes, and add 50 microliters of protein kinase inhibitor to each tube. Vortex each tube and incubate the samples at 37 degrees Celsius for 30 minutes.
The correct amount of titrated Peptide-MHC tetramer must be diluted in enough master mix to stain all of the experimental tubes. Therefore, it is important to prepare a 15% excess of the master mix to account for pipetting errors.